let's look at our answers for what does the human genome have been sequenced mean. So somebody said that the genes that make humans human have all been identified. This is actually our goal, but that's not what it means. So I'm going to parse out each part of this sentence because this is actually a common thing that people think. So a gene is a sequence of DNA that encodes a functional molecule. So the more often, the more common functional molecule that it's encoding is ultimately a protein. It's not always protein, but if you remember, or I don't know if everybody knows about central dogma of molecular biology. If you don't, you're going to see it here. Genes are DNA. They get transcribed into RNA, and then that RNA, if it's a messenger RNA, they need to be translated into proteins. And then the protein is the active part of the cell. So active as in it either does some function, so it's an enzyme, or it's a structure protein, so it's a building block of the cell. So the genes are DNA that carry information on how to make proteins. Some genes don't carry information to make proteins, so those are the exception. They're called RNA genes, which means that DNA gets transcribed to RNA, but it's not mRNA. It's either tRNA, which is transfer RNA, or rRNA, which is ribosomal RNA. So those are still functional. They do stuff in the cell, but those RNAs, they do not get translated to protein. But the genome, which we'll see what that means, the genome is more than just genes. The genes are the parts that encode those functional molecules, either ribosomal RNA, tRNA, or mRNA that would be translated to protein. There are other kinds of RNA too, but these three are the main. And let's see. Yeah, so let's see. I guess we parsed enough on so the genes, they are part of the genome, but the genome has more things beyond the genes. It also has DNA that does not encode anything, that does not become any kind of RNA. Okay, so, and then to make humans human, or any living being, whatever the living being is, that does have to do with the genes. That part is true. Okay, so all genes in humans have been discovered and put in order. Okay, the put in order is probably the better part. We are still like discovering, as in, you know how I mentioned here that the genome has genes and then other part of DNA that's not a gene, meaning DNA that does not get transcribed. So when we say the genome has been sequenced, then we actually, the answer to that, which is what you want to hear, is that we have all the letters of the genome, but we actually don't know what those letters do. We don't even know if they are genes or they are not genes. I in a moment will show you an example, like an analogy to what that means. So when we say the genome has been sequenced, it means we got the letters, but we actually don't know if they are genes or not, so we don't know that the genes have been discovered, but we potentially can find the genes, right? Because it's like you have a book that you now have access to, and then you can find in that book the words, which would be the genes. So yeah, and that this placed in order, what I meant is we know the order of the letters, and the letters for DNA are either A, T, C, and G. So this person here wrote exactly what it means. And yeah, so sequencing means we know the sequence of A, T, C's, and G's. That's what genome being sequenced means. But like I said, we don't actually know what they do. Let's see. Genetic code is something else. you're going to learn to use the genetic code when you're translating things. So the genetic code is the 3-letter DNA that gets transcribed to RNA. So those 3 letters, they're called a codon. Which amino acid do they encode? So if you Google genetic code, you're going to get to a table. The genetic code is not the genome. The genetic code is when you have these 3 letters in sequence, which amino acid are they encoding? So that's something else. We'll also see that later in the semester. Okay, so a lot of people are saying things about genes, so I'm glad that we are going to do exercises on, once you have all the letters of the genome, how do you find the genes? So, because there are bioinformatics tools for that, but my main goal when I show you that is to show you that what's the composition of a genome, and then you'll see that genes are just a part of it, and it's actually a very small part. So, yeah, when you say decoded, maybe you meant we have the sequence letters. Okay, so another person said that. And then I'm just going to browse through all of those. But when we say the human genome has been sequenced, it means that we know the letter orderings. And when I say human, then specifically for human, but any genome can be sequenced. So like remember COVID got sequenced. So even though COVID is an RNA virus, okay, we got to the sequences of A, U, C's, and G's. So the COVID genome has been sequenced. Now we know the letters, the A, U, C's, and G's, how they appear in this genome. So yeah, that's one of the things I wanted to show you. So the difference between sequencing and mapping we know, so there is some chunk of sequence. We know in which part of the genome it shows up, which is basically the order. And before we could sequence genomes, we could already map things. That's what you do in genetics. I don't know if the word centimorgans ring a bell, but when people were mapping the genes, we were doing this before we even had the ability to get all the letters. When people were mapping genes, they were looking, this one gene is how far away from this other gene, and that was all done through statistics. So people knew that if they're very close together, then it's unlikely that you're going to have recombinants. If they're far apart, then you're going to have lots of recombinants. If you don't know what this means, don't worry. It's not important for genomics, but you're going to learn that in genetics. And if you don't learn that in genetics, that's fine. But what I wanted to say is that mapping is knowing the location of something, and used to be the location of something in relationship to something else, because we didn't even have the sequence. Yeah, I don't want you to have a strong grasp yet, because otherwise you wouldn't be in this class. So thanks for, you guys are so good and humble. Like, I like when it takes strength to say what you don't know. It makes me happy. Yeah, so we don't know function yet. We're going to establish that later. And let's see. So we don't know if the DNA is a sequence, is a gene. We don't know if it is a gene. We may not know what it does. Maybe we have a hunch that it's a gene because it has some cues that will make your gene predictor say it's a gene, but we may not have any idea of what it does. And then where the genes are located, that's mapping. So no, the order is mapping, and then the genes are the DNA sequences that are transcribed, so into some sort of RNA. Not that one. Let's see. Yeah, so this part is the right part on sequencing. The chromosomes are much they are actually visible under a microscope. People could identify chromosomes just looking under the microscope, but you cannot see the DNA sequence through the microscope because that's too small. So chromosomes were identified, I'd say, at least as early as like the 30s, maybe. I have to look that up. But if when people would stain a cell, when the cell is in division, then they could look at chromosomes. Maybe it's even earlier than that. And now I'm curious, but I'll look that up afterwards. Okay, again, genetic coding. You can Google that and see. You're going to find a table that will show you these 3 DNA sequences or RNA sequences will encode this one amino acid. So it has to do with the genome, but genetic coding or genetic code is actually specifically used for this. These letters encode which amino acid. Okay, so yeah, the order of nucleic acids, I guess you mean the A, T, Cs, and Gs. Yeah. So this we know all the genes. Okay, we don't actually know the genes. So for example, when we sequenced COVID, then you first obtain the sequence and then you get all the letters. And now you're going to go look for words in that sequence, and the words meaning the genes, by comparing to words we already know from other viruses. All right, so I think I went over all of them. I'm going to go back to the PowerPoint presentation. And then some parts maybe already became clear because I repeated them. And then other parts become clear when I show you in the slide. Okay, so here the question that I've answered a few times, but now it's going to be a little maybe more graphic. So what is genomics? So what? Yeah, it's the study of genomes. And like, oh, okay. So what is a genome? So there are, as I said, it depends on how pedantic you will be with the definition of genome. This is the more pedantic one, which is it's the haploid DNA content of an organism. But oftentimes people don't think that specifically, right? So whenever we think we sequence some genome, all we care is that we have one string, like one version of all the letters that compose the genome. But if you remember from genetics or from like 211 lab back in the day, you have, if you are diploid, so diploid is you're not haploid. Haploid, there is it's only one part that you receive from one parent, but biologically you have 2 parents. So you receive one genome from mom, one genome from dad, which makes your one genome, but that's not your haploid genome. That's why there are more definitions, right? So the pedantic one, as I said, is the haploid DNA content, which then, depending who you talk to, includes mitochondrial genome. And other people say no, genome is specifically for nuclear. But in any case, just think the genome is all the letters that are inside of your cell of A, T, Cs, and Gs in terms of DNA. And if you're an RNA organism like a virus, then your genome is all the letters in the RNA that you're going to use to process information. Okay, so the second definition is the complete set of genes or genetic material present in a cell or organism. So I kept telling you that it's not just genes. So that's why this says genes or genetic material. Genes are DNA that gets transcribed into some sort of RNA. So ribosomal transfer, messenger RNA gets translated to protein. So Genome is genes plus other stuff. So this other genetic material is all the DNA that sits there and does not get transcribed, doesn't become RNA. So people used to call that DNA junk DNA, but now we're learning what it does. Okay, so I threw the definition haploid there. So if you don't know what that means, don't despair. Here I'm going to show you. It's the number of chromosomes in a gamete. So a gamete is one of the parts that will get together with the other gamete and then make a new living being. So if you are a flower, you have your ovule, and then when the pollen comes in, so the ovule has its own haploid genome, and then the pollen fertilizes that ovule, and now you have something that is diploid because it has the haploid genome of the ovule, the haploid genome of the pollen, and you have a full diploid genome. So that's actually the simple case. And we are like that too, humans. There is egg for the woman, sperm from the egg. The egg has a haploid genome. The sperm has its haploid genome. And then when they come together, they will make an organism that's not haploid. Okay, so I said that that was the simple case. When it's haploid, no, 1 plus 1, it's 2. So we have 2 versions of every chromosome, right? We got 23 from mom, 23 from dad, and now we have 46. But if you're interested in plants and some animals like salamanders or whatever, then there are many more polyploid plants than animals. But so you don't know this, and this is more like curiosity, and mostly because that was the topic of my PhD dissertation. So when you think about polyploids, then you're like, what does polyploid mean? So poly is for many, right? And it means that you have more than 2 copies of everything. So let me show you graphically what does that mean. I'll compare a diploid with a polyploid. Okay, so let's see here. This is you, right? You are diploid, and we call that you are 2x. The x meaning it's the monoploid number. It's how many times each chromosome shows up. So here's chromosome 1 from mom, chromosome 1 that you got from dad, chromosome 1 from mom, chromosome 1 from dad, or vice versa, I don't know. So this is you. A somatic cell, which is a cell from your body that's any cell that's not a gamete, 46 chromosomes. We have 23, and then 2 copies of each. So when you're haploid, then you have half of that. So if you have a gamete, either sperm of egg cell, then you're haploid. The haploid has 23 chromosomes and 1 copy of each chromosome. That's a simple scenario. That's the most common scenario. And the next scenario is mostly curiosity, because I want you to know that such things exist. And it's pretty amazing. That's kind of how we got seedless watermelons, which is like why I thought those genetics are so cool. Okay, so one definition, genome is the haploid DNA content. So if you look at this, in one definition, half of these chromosomes is 1 genome. In another definition, this whole DNA here from all these chromosomes, that's a genome. When you're sequencing, you're reading all of the letters from this beginning of this tip of this guy all the way to the end of this, if you're a guy, right? So here's the complicated scenario, which I'm bringing up because I said it's, biology is so interesting. Not everybody is diploid. Like strawberries, which were I was sequencing when I was doing my PhD, they are called octoploid. So they have 8 copies of each. So I just took this like human karyotype here, and I made copies. What if the chromosomes from strawberry looked like that? So this is what the strawberry nucleus would have inside. So for chromosome 1, they would have 8 copies of that. For chromosome 2, it would have 8 copies of that. And strawberries only have 7 chromosomes. They don't have 23 like us. They only have 7. But for each of the 7, they have 8 copies. So your somatic cell has 56 chromosomes, and there are 7 chromosomes, 8 copies of each. So what do you think? Actually put on the chat. If you go with the haploid definition, which is the whole reason why I brought this, this graphic here. If you think haploid is the DNA content of a gamete, how many chromosomes do you think that a haploid, so the strawberry pollen, how many chromosomes are there in the strawberry pollen? I'm going to minimize to look on the chat. So there are 7 chromosomes, but guys, good job. Yeah, 56 divided by 2. Good job, yeah, 28. So the reason why I presented this is so that you know that haploid doesn't necessarily mean 1 copy of every chromosome. It means that for whoever is diploid, because you have this and then you only have 1 copy of this. If you're polyploid, then you're going to have 1/2 of whatever is in your somatic cell. So strawberries have a huge variation. And I have a friend, we shared a lab bench and an office, and he's the head breeder for Driscoll's, you know, the strawberry company. And everybody buys strawberries. I try to buy Driscoll's because, you know, I'm supporting my friend. Anyway, he basically he breeds them, and there is so much variation because, can you imagine if these guys are heterozygotes, so like if there is one version of a gene here, a different version here, a different version here, a different version here, can you imagine how many combinations you have? It's like you have a deck of cards, and for every card, like an ace, instead of having 4 variants, you know, the spade or gold, whatever it's called, the hearts, you'd have 8 variants. So you have so much more variation. It's pretty amazing. So I don't know, I just like presenting amazing stuff. And you'll be like, oh, yeah. So yeah, in the somatic, you have 56. But in the haploid cell, you have, okay, this is covering, but it's 28. So you're going to have still 7 chromosomes, but 4 copies of each instead of only 1, as we do. And then that generates the question, if the strict definition of genome is the haploid genome, then how many copies are there of each chromosome? Oh, for the strawberry, there are 4 copies of each of these 7 chromosomes for the haploid genome of strawberry. So then we need to say, okay, but what if I don't want to refer to the strawberry chromosome by the 4 copies of each of the 7 chromosomes? Because when I'm sequencing strawberry, I really only want one sequence for chromosome 1, and there will be some variation. But if you get 1, then you have a very, very good idea. I want 1 sequence for this. And truthfully, when we are sequencing, these all get like scrambled. But with what we know or what we want is to get the full sequence of chromosome 1, the full sequence of chromosome 2, blah, 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 full sequence of chromosome 7. So when you're referring to, I only want to refer to the number of, I want 1 of each chromosome, that's called the monoploid number. So yeah, so when we sequence the strawberries, we want to sequence enough DNA to sequence 7 chromosomes, which would be sequencing the monoploid number. Okay. Now, I wanted to know that there are organisms that are not diploid, but I wanted to know haploid is the number of chromosomes in a gamete, and for polyploids, the haploid number is different. It's not just one copy of each chromosome. And the word monoploid here is mostly curiosity. It's just that I also deep into this stuff that it's hard for me not to tell you about it. Okay, so I told you that Genomics studies genomes, and you're like, okay, what kind of studies can you do? So one of them is sequencing DNA. That's the basic one, and now it's done very, very easily. When I go over the history of sequencing the human genome, you're going to be amazed on how much time, effort, and money it costs, and how easy it is today. It's a victory for science. So we can do something called fine-scale genetic mapping. So you're like, what do you mean? I said, okay, mapping is you are looking in the in which chromosome does this piece of DNA happen? So that's the localization of that chunk of DNA. In fine scale, it means you know very precisely where that piece of DNA occurs. While in genetics, you have an approximate location in comparison to another chunk because you've determined that by statistics. But in genomics, we have the exact DNA sequence, so we know exactly in which letter a gene starts and the next gene, or where that one gene starts and stops. So we may be wanting to look at the different DNA sequences in a population. Many reasons why you'd want to do that. There'll be classes just about that. Measuring transcriptional control, and maybe you're like, what's that? So I told you that genes as DNA sequences that get transcribed. So there's an enzyme called RNA polymerase that reads the DNA and makes an RNA version of that DNA. So that process is called transcription. Transcriptional control means that we are trying to decide when is that DNA going to be transcribed to RNA. The DNA still exists. When I say become, I don't mean that it shifts. But when is that DNA molecule going to produce an RNA molecule of it? And also study interactions. So here's the next Google form. And then I will do a demonstration, and that will be that. So, or maybe, yeah, I'm going to pair you guys so that you can try to figure out what does this mean? What does this mean? You're going to try to make relationships, and then I will answer that, and that's going to be that. So let me break out the rooms so that you can talk with each other. So maybe say hello, introduce yourself, and then say